All right. Hello, everyone. Welcome back to the channel. I'm your host, Zach Hancock. I'm an evolutionary biologist who specializes in population genetics, and I'm really excited for the discussion tonight. Something has happened that I never expected to see. A creationist gave a talk at the evolution meeting. Now, this is not like a biophysics meeting that has a little evolution component that they could sneak into, or like a creationist meeting that just has an evolution title. No, this is the evolution meeting, the actual one. We are really, really excited about this. I am joined by a great guest, Dr. Dan Cardinal from Creation Myths. Dan, go ahead and introduce yourself.
Hi, I'm Dan. My channel is Creation Myths, which you can read right there. And I can never do the mirror thing. On Creation Myths, I try to address the scientific claims of creationists and explain why they're wrong in relatively short snippets that are understandable for like non-biologists is the goal. Make it really accessible for everybody. And also deal with all the kind of related pseudoscience and like right-wing nationalist politics that comes along with creationism. So that's kind of my wheelhouse is the science, but then also, hey, here's all this other stuff that comes along with it that we need to deal with.
Yeah. People probably saw in the description, we were supposed to be joined by Dapper Dinosaur. He unfortunately couldn't join us today, but we will definitely have that dinosaur on the channel eventually. So we're going to be watching the talk today that was given at the evolution meeting. But before we do that, we wanted to talk a little bit about the evolution meeting itself, like what it is and why it's like such a big deal. So the evolution meeting is actually the oldest meeting that is hosted by a society that specializes in evolution in the world. It's hosted by the Society for the Study of Evolution, that's SSE, that was founded in 1946 by Ernst Mayr. And so that was the very first, like in the whole world, the US had the very first evolution society. So that's pretty exciting. And this is their annual meeting. It's actually a joint meeting between SSE and the American Society of Naturalists, who run the American Naturalist Journal, and the Society for Systematic Biologists. So the three of those societies, they get together, and then just like thousands of evolutionary biologists all around the world just converge on some city every single year to present their work and talk about evolution. Basically, all the big names in evolution are there, and then some of us much, much smaller names also go and present our work and talk and get to meet all of the big name people. This is the meeting that I go to, that I present at. I present at it almost every year since I started going in grad school. I'm not going to go this year, but I did wear my Providence 2019 t-shirt from SSE to sort of celebrate the occasion today. I know, Dan, as we were talking about before we joined the stream here, that Dan also went to several of the evolution meetings as well.
Yeah, I went to I want to say 2012 in Ottawa. And then I actually just looked it up. The one in Chicago that I was thinking was not an evolution. It was a different organization that did Chicago the following year. It was very similar vibes and very similar group of people, but that was not actually Evolution 2013. But the one that I went to was in Ottawa, and it was just a freaking delight. And my highlight, so I went to that meeting alone. I was a scared little early career grad student at that meeting. It was my first like big meeting that I went to. I went alone with nobody else from my lab. It was my first big meeting talk. And then during the poster session, I literally, and I mean this with no figurative exaggeration at all, I literally bumped into Richard Lenski. I literally walked into the man and said, "Oh, excuse me." And then his name tag was right there. And then I like fanboyed out a little bit, and it was really exciting. And he was very nice.
Yeah, I was very excited. I didn't actually meet him in person because it was, during the pandemic when I was at Michigan State. But I did meet a lot of members of his lab. And yeah, so that was really cool to like be in the same university where the long-term evolution experiment was going on. Yeah, that was super cool. So one of the things that I wanted to talk about is like this creationist presented at this meeting, right? Presented at the evolution meeting. And that's pretty exciting. So the creationist in question is one Salvador Cordova. Those of you that are familiar with me and Dan, you're probably also familiar with Sal. He's been around for a long time. From my understanding, and Dan can like definitely give some more context to this, but my understanding is in his Reddit days, they called him Slimy Sal.
Just to be clear, that predates the Reddit days. That goes back to the blog war days of the early aughts when it was like Uncommon Descent and Panda's Thumb going back and forth. Yeah, that goes way back. That was literally over 20 years ago.
Wow. Okay. So Sal has been around for a really long time.
For a long time in this. Yep.
Yeah. My familiarity with Sal came through John Sanford and his work with John Sanford. I don't know if Sal got his start there. Dan, if you could fill in some of the early details.
That was his kind of transition into biology, is my understanding. So he was more of a physics guy, and then he crossed paths with, interacted with, and started kind of collaborating with John Sanford of genetic entropy fame. And he kind of crossed paths over into the bio side of things at that point.
Okay.
And Sal, feel free to correct me if I'm wrong, but that's my understanding of things.
Yeah.
As I know it.
Yeah. So Sal's got a couple of things that he's really big into. The two things that come to my mind are like the protein orchard argument, and this is the idea that since proteins don't have universal common ancestry, that somehow disproves universal common ancestry, when no evolutionary biologist has ever argued that.
Nobody thinks that's the case. In fact, the opposite is the case, because de novo genes are a thing.
Yeah, like you have intergenic sequences that are not protein coding that then become protein coding.
Right.
Therefore, it has no ancestry with other protein coding genes.
That is a necessary process in evolution, and we can very clearly document that happening in genomes. That's the thing that happens. So, yeah.
Yeah, but it's a thing that he's been on for a long time. And then another thing that he's been on is what he's actually going to give his talk on, and that is on the concept of fitness. So we're going to spend a bit of time today listening to Sal just butcher the concept of fitness, and then we're going to try to correct the concept of fitness. Yeah. But one of the things that you might be wondering is, how in the world did a creationist get a talk accepted at an evolution meeting? How do they actually accept the talk and allow him to give this talk? And so I wanted to just kind of talk through how this happens, right? Like how you submit a talk to a conference.
Well, Zach, so the first step is you get that little when you do, I guess it's not in the mail anymore, but I assume back in the day it was in the mail. But now it's just an online waiver that you fill out, and it's like a little checklist, and you just it's like AIG statement of faith, but you have to check all the boxes, right, about accepting Darwinism, and then you have to digitally sign at the bottom, and then as long as you sign that document, then they'll consider you, right? That's how it works, right?
Exactly. Yeah. This is why I wish Dapper was here to give the prayer to Darwin thing that he has. It would be perfect to insert right now. But yeah, you have to say a pledge to our Lord and Savior Darwin. No, the only thing that you have to do, and the evolution meeting is actually pretty accepting in that you don't have to have any affiliation with the university at all. You don't have to have any kind of academic affiliation or credentials for that matter. And they base it on a first come first serve basis. So really all you have to do is pay your registration. If you pay to go, and then you submit an abstract. And your abstract just basically can't be crazy. It just can't be something violent or racist or it has to be about evolution, and that's it. That's really all you have to do, and you have to get it in time that you're not falling to the bottom of the barrel, and then you will have a slot to present your talk. And this is why Sal was able to do this. All he had to do was submit an abstract. He put in his abstract that he was going to talk about fitness and biophysical limits to fitness, and they let him give the talk.
Right.
And that's literally it. That's all creationists. That is all you have to do.
Every one of you out there, what's your excuse? All of you.
Yeah.
Zach froze. You're back. Yeah. Everyone else out there. That's it, guys. I do want to say, I'm going to take a little bit of credit for this, because I've been saying to every creationist I talk to for years, look, you think you got the goods? Publish, go to a conference. I'm not saying you have to give a talk, just do a poster at a conference and just put yourself out there. I've been saying this for years. I've said it to Sal, and he did it. And you know what? Whatever you think of Sal, whatever you think of the merits of the presentation we are going to watch, you know what? Credit where it is due. This is somebody who put his money where his mouth is, something that no other professional creationist has done to date, right? You can go back to the 90s with the ideas. You can go back to the 60s with the original, creation science people. None of them are actually trying to engage with the real scientific community here. Where's Nathaniel Jeanson on this? Rob Carter, Sarfati, Jeff Tompkins, right? Great example with the human chimp stuff. Dude, put it out there. You got the goods. Like, bring it to where these conversations happen and do it. Nobody has done it except now Sal Cordova. So, like, the rest of you, Michael Behe, you know how science works. Get in there. Come on. Like, put yourself out there. And they just won't do it. They will not do it.
Yeah. So I just wanted to put this comment up from Rebecca. Hi, Rebecca. Thanks for being here. That was a joke. You don't actually have to sign anything.
I was joking. That was not real.
You don't actually have to sign anything. You can go to one of these conferences and say you hate evolution if you want.
Yeah.
There's no statement of faith. There's no statement that you accept evolution, because Sal was able to present, right? So very clearly, Sal did not, I assume, and we're going to watch his, talk. So you're going to see at no point in his talk does he say, yes, I accept evolution, right?
And several times, if you know the lingo, he drops dog whistles to the effect of, I do not accept evolution.
Yeah.
Several times. Yeah.
So no, I agree. I think that, creationist, this is your opportunity now. Now you see that someone has done it before you. And what Sal did was actually like a pretty low stakes way of doing this. So the evolution meeting historically has always been an in-person meeting, but ever since the pandemic, it's been split into 2 meetings where you've got like a month before the in-person one, there's a virtual one, which is like really low stakes. You just pop into a Zoom room, you give your talk, and you pop out, and you can basically avoid, anybody like following you afterwards and asking you a bunch of questions like maybe they would in the in-person one. Because let's be clear, Sal, if you had given this in person and I was there, we would have been talking in the hallway.
Would have had a conversation about fitness.
We would have had a conversation at the poster session or something. But that's fine. That's fine. So this, you can do it in the virtual meeting at least, which is a much lower stake. I also think it's maybe cheaper than the in-person one. But yeah, this is your opportunity, guys. Like you can do this. Sal did it. Go forth and present your work to the rest of the evolution crowd. Okay, so
do it. Bring it.
With that said, the kind of plan that I want to do here is it's only like 13 minutes. So it's not very long. And so we'll be able to listen to and react to the whole thing. And then as he's going through, we'll pause, we'll discuss, and we'll sort of chat through it. He's going to bring up a series of papers that he brings up all the time. There's like 3 papers that he discusses in every single talk he ever gives. And I think it's maybe worth it for us to go into them and sort of like talk about them in a little bit of detail to sort of clear up what they're about, because he just constantly misrepresents them. So I think it will really, really be good to dig into them a little bit. But yeah, and then we'll talk about what fitness is and why it matters. And yeah, so I'm pretty excited to hear what Sal has to say here.
Before we get into the talk, I just want to context for everybody here. So the whole point of this talk is, and I will say before I explain this, it took me a couple listens to get the point. It took me a couple tries. But this is, if you've seen Sal do stuff on YouTube, a lot of what he goes through here is going to be familiar to you. But basically, this is about how the concept of fitness, that is how we define fitness in evolutionary biology, related to reproductive success, or as I like to explain it, the representation of your alleles relative to everybody else's alleles in subsequent generations, how I like to do it. Basically, that is not a good way to describe fitness, and it should be described in terms of like biophysical characteristics and engineering, fine-tuning, or whatever, right? Complexity in some way. And I've had conversations with Sal about like how he wants to define fitness, because this is also the topic of the book chapter that he co-authored with a bunch of people. It was Ola Hoster and Bill Basner and John Sanford. I forget if there are any others on there, but those are 3 I can remember. And then it's also, that book chapter was basically an expanded version of the 2018 Basner and Sanford paper about the limits of fitness improvement in populations. related to Fisher's fundamental theorem.
Yeah, the idea was about like and he's going to talk about this as well. And we can get into the details, but it's about like their critique of like maximization principles.
Hey Zach, has anyone directly addressed that paper at any point in history? Just so we're clear, like whether this argument that Sal is building off here, whether anyone in the peer-reviewed literature has, I don't know, taken it upon themselves to mathematically show that they're wrong.
Yeah. I think just like fairly recently, right? Like, or wait, how long ago was it now? Was it a year ago?
It was over a year ago that paper was published.
Wow, yeah. Okay, so
time flies.
Over a year ago now, with I want to say crickets as response.
Crickets.
Nobody, which has been like kind of disappointing, honestly. Like when they published that paper, like it was all over the place, and like when we did our review of it, there was like a gazillion people that covered it, and like we have received crickets since we published that paper.
It's creationists don't want to deal with it, but anytime any creationist brings up the Basener and Sanford fitness stuff or genetic entropy, because you showed, using empirical DFEs that genetic entropy is wrong, anytime any of that stuff comes up, I'm just like, here's the paper, respond to the paper or don't waste my time. Like,
okay, let's get started. Let's hear what Sal has to say. This should be a lot of fun.
Worst possible time my slide presentation fails. Okay, on behalf of myself and my co-authors, I extend thanks and greetings to participants and organizers of Evolution 2025. My lead author, Bill Basener, is an emeritus professor of mathematics. John Sanford is a retired Cornell research professor and genetic engineer, famously known for his invention, the gene gun, which, by the way, is in the Smithsonian National Museum of American History. Ola Hosger is a professor of population genetics at Stockholm.
Hang on. I just want to point out that line, the Sanford inventor of the gene gun, which by the way is in the Smithsonian, that is one of like the Sal Cordova bingo / drink lines.
Drink.
Like, so whatever you got, that's whichever game you're playing, that's your cue right there. Yeah.
That is such a funny line. I have some things to say about this intro. I'm gonna let him do it.
Yeah, sorry to interrupt on that one, but let's.
University of Stockholm. I'm Salvador Cordova. I'm presently a PhD student in biomolecular engineering at Liberty University with 5 previous science degrees, including a master's degree in applied physics from Johns Hopkins University. I was previously a senior engineer and scientist in the aerospace and defense industry working for MITRE, before getting retrained in biology by John Sanford and studying at the FAES graduate school at the NIH.
Okay, so one of the things that you don't have to do when you give a talk, and maybe I should say this as the normal structure of talks at conferences, do not involve reading your CV before you give the talk. Like you don't have to say, like normally what you do is at the very end, you have a slide of like an acknowledgement slide with just like the people that helped you, your funding sources, and that's basically it. Like, you don't have to say, I have X number of degrees from X universities, and I worked with this guy whose invention is in the Smithsonian Museum. Like, you don't have to do that. That's not like, and nobody does that. It's kind of a weird intro.
It's so the video that we're watching here actually has multiple talks after it. And I want to encourage everybody to just like, you don't have to watch all of like every minute of it, but just like skip through, watch the first 2 minutes of any one of the other talks in this session and just compare and contrast.
Yeah. It's so funny. I even like went back to watch because some of my talks, like actually both of my evolution talks are on YouTube, published from the same thing. So like people can go see them too. And I went back to just like compare it side by side, just because it was just like so weird. Another thing that like what we're not going to see here is like basically any data or any graphs or any like methods slides. There's nothing like that.
Nothing.
It's so weird. It's the design of it is very strange.
Which again, I had to watch this a couple times to like kind of follow it through. But then also, if you watch the other talks in the session, the contrast is very clear. It's all, as we will see, there's no primary data here, right? It's this guy said this paper says this, here's my interpretation, yada, 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 right? It's not structurally, it is odd for a conference talk.
Yeah.
We report on ongoing work that builds upon our previously published works in population genetics. protein biology, structural bioinformatics, and unpublished works in biophysics. So what brings me here today?
Wait, hang on, Zach. Can you go back just a second? Did I miss that? Because I was... Oh, it has Basner and Sanford right there. There it is.
Yeah.
I missed that because I was listening rather than watching. Yeah.
Yeah.
You got it right there. Okay.
Yeah. Here's the Basner and Sanford paper is indeed cited here, which is interesting because he's not actually on this paper. So I don't know why Sal is like throwing this paper up here
because he did that book chapter with Basner and Sanford, and so by the transitive property,
he wrote this paper.
he like this is part of his thing.
Yeah. He can put this on his CV by the transitive property.
And unpublished works in biophysics. So what brings me here today?
I just want to point out. Okay, so we're 1 minute and 11 seconds into his talk. This is he has 13 minutes to give this talk.
It's tight.
He has 13 minutes. Like everybody in the conference is only given. You're technically only supposed to speak for like 12 minutes and then give like 2 to 3 minutes for questions. Nobody ever does. Everybody always runs it all the way through. It drives me crazy. But no, everybody's really bad on time management in these talks. But he spent the first minute of his 13 minutes reading his CV. Like we just now got to the question that he's going to try to answer.
And just to be clear, like when you give a conference talk, like there's different styles of science communication. Like if you're teaching an 80-minute lecture in college versus giving a 12-minute conference talk versus doing like a popular science thing on YouTube, right? Those are all very different fora. They're very different styles. There are very different constraints you have. Like when I prepare for, like I tell my students, I never give the same lecture twice. I never practice. What you see on the slides is what I'm working from. And like, I'm improvising mostly. I'm not word for word going through a script. When you do a 12-minute conference talk, it is completely different from that. You practice it over and over and over, and you get it super tight, and you have to hit your marks at specific time points to make sure you get through what you want to get through. And nobody is going to waste the first minute on biographical stuff. Like there's a program, people can look you up. Nobody's going to waste that first minute. It's just, you're just giving away time that you could use to explain whatever it is you want to talk about. It's weird. I don't know.
Yeah, it is really strange. And I'll say just like from my experience, I would always like practice the talk that I'm going to give a gazillion times such that by the time I give that talk, it's almost identical to what I'm practicing, right? Like there's nothing off the cuff. But what's interesting is that I'm pretty sure Sal is reading from a script.
He is. He said that in another video. Yeah, because he did a separate video on his own channel where he basically re-gave the talk. And he said in his little intro spiel to that aside from a couple of ad libs, it's basically identical because he's just reading what he had prepared. So, and this, which you can't do that if you're in a room full of people at the physical conference, but online you can have tools and you can just read through it.
So which means that like he could have written this such as to have given himself, another minute to make his point instead of spending it on biographical things.
Yeah.
All right. So we are now 1 minute in and we are getting to the problem.
This comment by Richard Lewontin, who said, the problem is that it is not entirely clear what fitness is. And no concept in evolutionary biology has been more confusing. Further, he laments the incommensurability of the original Darwinian conception of fitness with the definition of fitness in modern population genetics. My goal here today is to add even more confusion to the issue.
I agree. And so what I want to do is I want to stop for a minute and maybe we can just talk about in a little bit of detail, just like sort of what fitness is, and in particular about this paper. Because those of you that have watched Sal for any amount of time are very, you've probably seen this paper before, honestly. So what I want to do is I want to just pull this paper up because I have it open in this other tab. We're just going to pull this paper up and then we're just going to like chat through it for a minute because I actually do think this is a really cool, interesting, and important paper. But the target audience is a little unclear from the way Sal presents it. So importantly, this is a philosophy paper. This is a paper that is published in the British Journal of Philosophy, right? Philosophical Science, right? So like, and now to be clear, that's not a dig. It's written by Dick Lewontin is on this paper, right? And he is one of the most famous population geneticist. So that's not a dig. I'm just saying the audience is very different.
It's a different, right. The objective is very different because this paper is not aiming to like rigorously quantitatively define fitness as you would in a population genetics context, right? It is a different, it's doing a different thing, which is totally legit and fair, but then you can't present it as this is intending to quantify this concept in a population genetics concept, and the authors conclude that that's not possible, right? That's a different thing.
Yeah, and I think like what Aryu and Lewontin here are sort of talking about is that among a lot of like biologists in this time, there was a little bit of confusion about what we meant by when we say fitness. And because fitness in a population genetic context means something different than what Darwin meant by it. And that's really what this paper is about. It's about drawing the distinction between these two things. And so they call Darwinian fitness the idea of like survival of the fittest, where fit in Darwin's conception is the relationship between the organism and its environment. It's like the environment is something that is perceived as independent of the organism, and then the organism has to fit to that environment. And so the better fit an organism is, or the variation among organisms in their ability to fit are these sorts of internal things that emerge, and then those that are more fit then, proliferate and then match their environment. And so that's the Darwinian idea. Darwin didn't actually use the word fitness. He used like fittest or fit, but it's kind of metaphorical, right? There's like this sort of metaphorical relationship between the organism and how well it fits its environment. This is not something you can quantify, right? Like this way of talking about, this way of talking about fitness is meant as almost allegorical, right? Like it's not meant to be rigorous and mathematical in the sense that population genetics uses fitness.
Can I give it like a silly example that I use in my head for this Darwinian concept of fitness, this organism matching environment concept of fitness? Have you ever seen, there's this wonderful video that, as someone who has young children who've had this toy, it's this great video of, it's a sorting toy with like blocks of different shapes, and you have the bucket, right, with the lid that has all the different shapes in the lid, and there's like a square and a triangle and a circle and like a bridge, and there's all these different shapes. There's this delightful video of somebody doing this and then a young woman reacting to it as they're doing it, and it's like the square goes in the square hole. There's like the circle block goes in the, that's right, the square, because they all fit through the square hole is the joke. And like that's how I think of it, where it's how well do you fit your environment, but it's not a precise thing. It's like, does your phenotype, like using modern language, does your phenotype compete effectively in your environment? And that might mean it's a triangle shaped block going through the square shaped hole, but it still works, it still fits in that environment, right? That's the silly analogy that I have in my head. It's that silly meme video that if you've never seen it, it's very funny.
Yeah,
especially if you have small children.
I think that does work pretty well in thinking about this sort of Darwinian fitness. But I want to see if I can just demonstrate for a moment what I think is the difference between these two things, and because I really want to drive home this idea that the population conception of fitness is actually different from this. So we're going to do just a tiny bit of whiteboard math here. And so what I want to do.
Hang on, time out. Hang on. For everyone playing along, Zach doing whiteboard math, bingo and/or drink.
Bingo. Yeah. So I think this is going to be a useful exercise, I promise. So just follow along. So we just talked about Darwinian fitness. And really, we're doing this because I want you to understand the Lewontin paper. I want you to understand what the argument is. Okay. So with that being said, we're going to start off. I lost Dan there.
No, sorry, I am here.
Okay. All right. So we're going to start off by thinking about the problem with Darwinian fitness is that it's not quantitative, right? You can't actually make predictions by it. You can't put numbers to it. It just doesn't work that way. But then come into like the rediscovery of Mendelian genetics, and what we found is that genes are these discrete quantities passed from one generation to the next, right? And they're faithfully passed on from one generation to the next. And so the population geneticists, what they wanted to do is they wanted to take Darwin's idea of evolution, Mendel's idea, which had these like sort of statistical properties to it, and then merge these things. But they had to operate on these discrete quantities. So let's say we've got two alleles, we'll say A, big A, and little a, and then those are at frequencies P and Q. Okay, so those are the two things. And then if you are a diploid, you have either big A, you are big A, or you are little a, right? So these are the three possible genotypes that you could have if you are a diploid. Now, the population geneticists are like, okay, how can we predict what these genotypic frequencies should be in each generation, right? If there is nothing else but Mendelian inheritance, if inheritance is the only thing at play, right, such that the alleles are following the law of like segregation, right? They're just segregating independently of each other.
Randomly going from diploid germline to haploid gametes with nothing else happening.
Exactly. And so then we can do something really simple. We can just say, okay, well, whatever the proportion of the big A, whatever the proportion of the little a, we can draw this little box, which I'm doing poorly, like so. And then we can just multiply it through, right? So P times P is P squared, P times Q is PQ, P times Q is PQ, and then Q times Q is Q squared, right? So then we can just write this out as P squared plus 2PQ plus Q squared. And since this is a frequency, it must all sum to 1, right? Okay, so there we go. There we have our distribution of genotypes. Again, P squared is AA, 2PQ is our heterozygote frequency, and then Q squared is our other homozygote frequency. Now, the question is, in the next generation, what do we expect that frequency to be? Well, again, if Mendelian inheritance is the only thing at play, then we just do this little exercise again, and we will get this exact same distribution again. So from generation to generation, the frequencies of the genotypes and by extension their alleles do not change through time. Now, if you are savvy in evolution, then you already know what I'm describing here. This is, of course, the Hardy-Weinberg equilibrium principle. This principle is super, super important to understand why we had to change the definition of fitness from a Darwinian idea to the population genetic idea, right? Because each generation, if the only thing at work is inheritance, the genotypes don't change through time.
Can I just say, Zach, the way that I love to, when I go through this in my class, and I just said in chat, hello, everybody in chat, delightful to see you all here. The way I describe this in class before we get into anything else is I basically just say, look, Hardy-Weinberg equilibrium is Newton's first law for genetics, right? Everything's going to stay the same unless it's acted upon by some force that's going to make it change, right? So if you don't have any of the things, then it'll just stay the same generation after generation.
Yeah. And if you don't know, Newton's first law is the inertia law, right? And in fact, there's like Elliot Sober, who was like a famous philosopher of biology, argued that the Hardy-Weinberg equilibrium principle is in effect akin to Newton's first law. That it
I just want to say, I came up with that independently and then later heard that, but I just want to say that I was using that in class before I knew that that was the case.
No, you know, convergence.
Convergent evolution.
Yeah. Okay, so what this implies then, what we then need is we need an extra term, right? We need something that will allow this frequency to change through time. Otherwise, it's not going to change through time. And the main thing that when we're thinking about these frequencies changing, what that means is that the proportions that they are in the present generation is different in the next generation, right? So something is biasing that proportion. And the way that you often do this in statistics is you add in what we would call a weighting term. So something that just weights those proportions from one generation to the next. And we will do this with these W's and then these little subscripts. The 1,1 is for the homozygous genotype, 1,2 for the heterozygous genotype, and then we'll do 2,2 for the alternate homozygous genotype. And now,
hey, Zach, can I just pause for a second and say that as someone who knows where this is going, this is phenomenal. And you should do a standalone video on this.
So I do have a video in which I derive this in a lot more detail.
Yeah, no, but just this though, just like I haven't taken a biology class since 9th grade, and just do like it. I just want to say this is great.
All right, I'm glad you're enjoying it. Hopefully the audience is enjoying it as well. So the idea here, right?
This is wonderful.
Is that each one of these terms are now weights, right? That are going to weight the proportions of the genotypes into the next generation, and you will in effect get change. Now, what are these little W's? What do they then represent if they represent a biasing in the proportion of these frequencies through generations? Well, traditionally, population geneticists have called this W term fitness. Now, what's really fascinating about this is that notice I have said absolutely nothing about organisms, about environments. I have not said anything about the fit of an organism to its environment. None of that has entered into this discussion. This is just a bookkeeping, right? We are just basically doing a sort of statistics here. And it's just demography. We are just bookkeeping each one of these quantities and how they're changing through time. And we call that differential reproductive success. We call that fitness. And now this is why we use this definition of fitness instead of the Darwinian definition, because this definition allows us to make quantitative predictions about how those frequencies change through time, whereas the Darwinian one doesn't, right? It's just a metaphor. And this is why this is so much more powerful way of dealing with fitness.
I just want to say that I have never, Zach, for all of the conversations we've had about this and for all of the videos of yours that I've watched, I have never seen you go through how fitness works at this like basic level. And I will tell you right here and right now, I'm going to steal that explanation from my class starting next year. I'm totally going to do that. That was way better than what I do.
Well, I hopefully, what I really hope that it draws home is this distinction between Darwin's idea and the population genetic idea.
It's great.
And this is what Lewontin was arguing. This is what he was trying to get across, is that you can't conflate the Darwinian concept with the population genetic concept, because and Elliot Sober uses this term as well, is that the population genetic concept of fitness is what he calls causally inert. It doesn't actually tell you anything about the cause of the difference in those proportions. Those proportions could be changing for many reasons. There's many reasons why PQ might be leaving, or P squared might be leaving more to the next generation than before. And then I'll just give you one last little thing that I think will be fun. And this is another little exercise that anybody can do if you're familiar with just like basic coding. This is the R statistical programming package or programming language. I'm going to use this little library called Learn PopGen. And then all I'm doing here is I'm assigning the different W's. So for that big A, we have a 1. For the big A, 0.8. And then for the little a, 0.5. So we will just assign those. Then we're going to give the starting frequency of P to 0.001. We're going to run our little simulation for 100 generations. And now what's fixing to happen is it's going to use that little equation that I just showed you. It's going to plug in those numbers and it's going to plot it over 100 generations. And what you get is evolution. You get the change in the frequency of P through time. Now, what's really super cool about this is because now we have a quantitative prediction and we can do two things. Let's say I could go out in nature and let's say I had just like survey data in which I saw the change in the frequency looked like this. But I didn't know anything more about the organism other than the change in the frequency looked like this. I could then use that curve and back calculate the fitnesses of the genotypes without having to measure them directly because I know what the change should look like. Alternatively, I could look at them in one generation, measure their fitnesses, and then forward project them using this equation. Right. So like this is what you can do with population genetics that you can't do with Sal's argument of what fitness is, as we're going to see here shortly.
I just want to convey to everybody. So Zach has gone through this. This has been a fantastic explanation of this. And I just want to convey to everybody, like, if you dig into this, it is genuinely like people describe math as like elegant math. This is beautiful, elegant science going on here. It is, you can use this in so many different ways. And, you know, in the evolution class that I just finished teaching for the summer semester that I teach, there are dozens of examples where we do this kind of thing, where we say, okay, here's what's happening, here's the dynamic at play, here are the predictions, here's what we find. It all boils down to a few very basic kind of equations that we can use to predict these things. And as you dive, you go further down the rabbit hole, the equations get increasingly complex, and there's different versions of heritability that come into play and whatnot. And it is genuinely beautiful once you kind of see how the different parts play a role. And I just want to encourage everybody watching to think about that, because Zach, I don't know if you recently saw, but a famous creationist, Dr. Michael B., he recently wrote a thing in which one of the things he said is that evolution persists as a field, essentially because biologists are afraid of math, are intimidated by numbers, and don't want to get quantitative, and therefore we don't have to critically evaluate our just so stories. And
so this is very funny. The same argument that Sanford made in his genetic entropy book, right? That everybody was just afraid of Haldane, right, and Fisher, and so they just wouldn't challenge their math because they were too afraid of it.
Hello.
Yeah. So I mean, what I've shown you here is just very basic. Like Dan said, we can teach undergrads this sort of thing. This is very basic, very straightforward. And if you're watching this and let's say you're skeptical, right? Good, because this is very simplistic. This is the most simple possible model. But if you stick with us, I can show you that these things can get a lot more complex, and we can actually account for a surprising amount of genetical evolution.
Don't make him get out the whiteboard, folks, because
don't make me do it.
Not joking.
Okay, so that's enough of an aside. I will stop that, and we will bring Sal back. So again, all of that just to put it back in context.
Zach, let me just, before we go back to Sal, I just want to kind of to reinforce what you just said, and to reinforce what PZ Myers just said in chat here, teaching this to undergrads, that graph that you just showed, I literally show almost an identical graph for that, showing the time to fixation for dominant, recessive, and additive beneficial alleles in the 200 level evolution class I just took. And we go through why those curves look different for the different types. This is not grad school stuff. This is undergraduate intro to evolution stuff.
Yeah. And the real key point to drive home here is that the whole reason I'm doing this is because I want to demonstrate what Lewontin was talking about, right? Lewontin was drawing the distinction between Darwinian fitness, which is metaphorical and not quantitative, and the population genetic, which is quantitative, but it has no causal link to it. It's not saying there's a cause to it. It's just bookkeeping.
It's agnostic as to why the thing happens. It's just describing the thing that happens. Yeah.
And so whenever you see Sal use that paper, you know that he's completely misunderstanding it and using it out of
Sal, do better. Listen to Zach. He knows what he's talking about.
All right, let's bring Sal back.
Answerability. Just kidding. Apologies in advance that I have to blast through these slides. Lewontin's lamentations are echoed to varying degrees. I list some here that can be investigated by further rewatching and recording of this talk in slow motion. Sorry, but we presenters are all under tight time constraints today. I recently queried Google Generative AI and asked, why is evolutionary fitness ill-defined? It responded, evolutionary fitness is an ill-defined term because it's a complex, context-dependent concept that's difficult to measure and define precisely.
Can we just time out?
Yeah.
You know who else you could have asked? Literally any evolutionary biologist.
Yeah. So
Sal,
I did.
You have my email. You could have asked me.
So of course, I also asked generative AI because the funny thing about generative AI is that it'll tell you anything you want to hear. It's all, and this is important, right? So you need to understand this about AI. All right, that AI is not about telling you facts or telling you truth. Right. It's about answering your question. All right, and it will do whatever it can to bend stuff around to answer your question. So all I did is I asked it, why is evolutionary fitness a well-defined concept? Instead of asking it, why is it an ill-defined? And look what it said. evolutionary fitness is considered a well-defined concept in biology,
exactly.
because it is fundamentally about reproductive success, differential contribution of genes, etc. While there might be nuances and different ways of measuring it, the core idea remains consistent and quantitative. So
AI is good now. AI is good now. That's it. That's all I got.
all you have to do is ask AI the question in the way you want it to answer, and it will literally tell you whatever you want to hear. So whenever I see creationists like use this AI slop on me, I'm just like, I immediately lose respect for you. Like do not do this.
I'm done. If I read, if I'm on, any forum where these conversations are happening and someone's like, well, according to AI, I'm like, I'm done. I'm not listening or reading to whatever follows that sentence. I'm out. No. I mean, I tell my students AI is just like stupid Google. It's a word association game. You ask it a question, it's going to find all the words on the internet that are related to that thing and spit out something in more or less coherent English. That doesn't mean anything that it spits out is correct, because it's a word association game, not like an encyclopedia with theoretically accurate information.
Yeah.
I hate it. I cannot believe that this man had the nerve to cite AI in a talk for the Evolution Conference. Sal, are you effing kidding me?
Yeah. That was also kind of a cringy thing, that you're going to read an AI generative response to literally like a Zoom room of evolutionary biology. It's
right.
just like,
and Zach, can I just get a time check on how far we are into this video?
We are 2 minutes and 20 seconds.
Okay, we're doing great. So if we extrapolate, folks, we're going to be here for approximately 5.5 hours. Hope you enjoy your stay.
So the fitness thing took the longest time. Hopefully we won't have to do anything of that extended pausing again.
Oh man. Well, I mean, you'll see. It's all, if you've watched Sal's YouTube channel, I don't know what the right word would be, obsessively as we do, then this is all going to be familiar.
Yeah.
Darwin wrote of endless forms, most beautiful and most wonderful, organs of extreme perfection and complication. Yet paradoxically he said, what a book a devil's chaplain might write on the clumsy, wasteful, blundering, low, and horridly cruel works of nature. For Darwin, the process of natural selection resolved this paradox. Darwin himself wrote, the presence of much suffering agrees well with the view that all organic beings have been developed through variation and natural selection. That said, it has become fashionable in light of recent discoveries in biophysics to say life is more perfect than we imagined, which is also the title of a frequent and popular lecture by William Bialik, a Princeton biophysicist and NAS member.
It's also very weird to like put up a seminar talk from just some random guy from like 2023 in your presentation at a conference. Like that's just like a weird, it's a weird choice, Sal.
It's a choice. It is a choice. And this is very clear creationist playbook kind of thing. Like, here's a smart dude in the field who said this thing that I can construe as supporting my position, right? That is a very common game that we play. And every one of the parts of that sentence, I don't know, sure, but like, smart dude in the field saying a thing that supports my position, you could dispute any one of those things, right? Except maybe the saying a thing. Somebody said a thing is hard to dispute. But other than that, you could dispute any part of that. And creationists love to play this game. And I will say, Sal he does it. He's very good at it. This is the game he plays, where he's going to quote mine the heck out of abstracts that he can construe as supporting his position. And here's an example, right?
Yeah, and this is, again, like in a normal academic talk, this would have been where you would be presenting data, where you would be like showing your study system or something, and like he's just giving quotes from people, right? Like he's just showing papers and giving quotes and showing people's pictures. Like, this is not generally how you discuss topics. You don't always have to have like original data, but like generally you would be talking about the science and not people and things that people said.
Yes, member. It is easy to believe biological systems are technologically inferior to human-engineered systems for various reasons. First, there is, as Darwin pointed out, lots of suffering, blundering, cruelty, and waste in the world. Second, human-engineered systems often outperform biological systems in certain, but not all, metrics. Third, A far more subtle but significant issue is something illustrated by Shannon's noisy channel coding theorem. That is,
do you know what he's talking about right now? I'm like,
he's giving a list of problems, right? One, life is bad and suffering and whatever, and it's more a human design can exceed biological design in some ways, and then also the noisy channel coding theorem, which can we all just appreciate that I'm pretty confident this is the first time someone has not only cited but also screenshotted and displayed a Wikipedia page at the Evolution conference, and I don't know whether this or the AI thing is worse.
yeah, I think that the only time I can imagine a Wikipedia page ever being screenshot in any other context is like ironically, right?
Right. But this is
it would be like as a joke, but like this being shown as like what he's just showing a big like noisy channel coding theorem. Ooh, and he's just showing the Wikipedia page for it. Like what are you even talking about?
There's
like,
here's in general. When you're doing a conference talk, because again, you are super time constrained, the key thing is efficiency. And Dr. Jay Bundy, for those of you that know Jay, Pocket Locker, this is something that he says all the time that I really appreciate, is that with science communication, it's all about efficiency. And in particular, if it's visual, there has to be like an ink to information ratio, right? And that is that you want to maximize the ratio of information to pixels, right? You need to show a lot of stuff very efficiently in a small number of figures because you don't have a lot of time. Yeah. And even if you're doing something looser like on YouTube, you still have to hold your audience. So you can't just be sloppy with this stuff, right? Yeah. So this is especially true in a very tightly time constrained situation like this. Am I supposed to read this? Am I supposed to listen to what Sal says? Because a rule of thumb, and this is just for everybody watching, a rule of thumb if you're giving a talk is if there are words on screen, your audience is reading the words instead of listening to you. And when they finish reading all the words, then they're going to check in and start listening to you again. So until they finish reading all those words, whatever you're saying might as well not exist, right? So everything you show on screen has to have a specific purpose that it accomplishes. What does this accomplish? And I know I'm being petty and nitpicky here, but what does this accomplish in terms of the context of a 13-minute conference talk? Nothing. He helpfully underlined the title of it for us, which great. Thank you. That's a cue that's what I should be paying attention to, but he blows through it so fast. He doesn't explain why it matters.
Yeah. Yeah.
So like,
and that's a consistent theme throughout his entire talk is that he's like throwing this is just a gish gallop. He's just like throwing a bunch of stuff at the wall and hoping something's going to stick. And the whole time I'm like, what does this even have to do with fitness? Right? Like your whole talk is about biophysical limits to fitness. And for some reason, I'm looking at a Wikipedia page for the noisy channel coding theorem. And I'm like, what does this have to do with?
You need to connect the dots explicit. You need to you can't just show the board with the red yarn. You have to put the pin and then run the yarn and then put the other pin. You have to walk the audience through it.
Yeah.
Communication systems that are optimized for compactness must, as a matter of principle, allow large amounts of errors to enter a communication and/or storage system that are later remediated by error correction systems such as Reed-Solomon and Bose-Chuduri-Hakogen schemes. By way of comparison, DNA has numerous ongoing breakdowns and errors, but DNA repair mechanisms correct almost all of those errors, and DNA is one of the most compact storage representations there exists on the planet. So, as a rule of thumb, the more complex, compact, and high-performing a system is, the more prone to breakdown and errors it will be. Hence, we can be misled to think an otherwise high-tech device is deeply flawed. With that in mind, here is one example of a feature in biology that was previously viewed
Can we just hit a timeout right here? Because he made an argument there without making the argument, but he did this like side swipe of the DNA is a complex storage medium, therefore design argument. We all know and love that argument. That's what he did there, but he didn't say any of those words. But that's the thing that he just did.
Yeah, I was very confused about what the argument he was trying to make there. Something like or how it was relating to Darwin's talking about the clumsy blunder.
I don't think it was. I don't think it. I think it was just a very basic, like, complex thing. Therefore, design. I don't think it's more than that.
Okay. All right. Yeah. Well,
I watched this talk 3 times, Zach. That's what I got. I'm doing my best.
I believe you. I believe. No, this whole like intermediate part of this talk was super hard for me to like.
Yeah.
What's the thread?
Now
here?
we're just in like examples though. So like good engineering examples, basically.
Yeah. Like he's literally fixing to do the vertebrate eye. You know?
Yeah. Here we are.
And blundering, but then later termed as ingeniously designed. The phrase ingeniously designed was used in a PNAS paper addressing specifically the backward wiring of the human retina. This is a case example of something previously viewed as clumsy and blundering, only to later be deemed ingeniously designed. As a former engineer, I love that phrase, ingeniously designed. Fasten your seatbelts in the interest of time. I now have to go into machine gun mode and fire off a long list of examples, which should be easy to track down by replaying a recording of this talk in slow motion.
Again, don't do this. Like, this is not how you give a talk. Like, never tell your audience, sorry guys, I'm going to have to talk so fast that you're not going to follow anything that I'm saying, but you can go back and watch it later. Like right. that is a bad way to give a talk.
Design your talk.
A couple of examples. Like,
design your talk to the time limit. You know the time limit. Design your talk to fit in the limit.
Yeah. Yeah.
Life is more perfect than we imagined, illustrating fitness through physics. Sorry,
I just wanted to say, hey, nice to see you. So I met this guy at was it Evolution in Albuquerque or Montreal? You have to remind me. But yeah, I came to my poster session. It's nice to see you. Thanks for being here.
Superior technological secrets have come to light from a deep-sea organism. Modern technology cannot yet compete with some of the sophisticated optical systems processed by biological organisms. Next, Bird Quantum Magnetic Compass. Bird Quantum Magnetic Compass operates at...
Sorry, I just want to tell Catholic traditionalists that Stephen Meyer could just like come to the evolution conference then.
Yeah.
Right? If he obliterates every prominent, evolutionist, why doesn't he ever come to the evolution meeting? He could come. He could give a talk.
No one's going to stop him. See my response, Zach. Put my response just right there. Yeah, I don't care if you're like on stage at Oxford doing a very formal Oxford debate about any of this stuff, or on YouTube for that matter. All the stuff that happens on YouTube is a sideshow. The actual debates on this happen in the scientific peer-reviewed literature and at conferences like the one we're talking about, right? Again, Sal and I, we go way back to like the Reddit days, like 10, 12 years ago. We've had a contentious relationship. I've talked to Sal directly about a lot of this stuff a bunch of times, but because he did this, he puts himself really above literally every other professional creationist, because none of them, with maybe one or two other exceptions people can think of, have done something like this, try to present at a real evolution conference, right? Any one of them could do it. Just like make it happen. Get in the ring, folks. If you got the goods, if Jeffrey Tompkins is so sure that the human chimp similarity number is south of 80% or whatever crazy number he thinks it is, bring it. Go to a conference and make that case. If Carter or Sanford or Jeanson or anybody else, if Jeanson is a great example, right? Because he said to my face that the mutation rate equals the substitution rate. Dude, bring that to evolution. Bring that to evolution 2026 and make your case that is actually happening in the mitochondrial DNA, right? Any one of them could do this. There's no ideological barrier to entry. None of them.
It's
Sal did. So
and the thing is you're going to be there with the people that actually work on this stuff, right? Especially with talking about the Tompkins stuff, that's the people that actually generate and create the algorithms for measuring and aligning genomes. They go to the evolution conference, right? So you will meet the people face to face if you are really willing to to do that argument. All right.
The quantum limit. Next. exciton transport is 100% efficient in photosynthesis. Which
No, it's not.
human
Nothing is
engineering
100%
can't
efficient.
ever
Let's just be
exceed in
clear
solar
about that. No, it's not.
Yeah, I don't. This is not even a citation, right? This is like the cover of a magazine or something, or a book.
I don't even know what that is. I don't care. Nothing is 100% efficient. Okay, it's just no.
It's like a perpetual motion machine.
Yeah.
I mean, it would be like a violation of like entropy.
It would be a violation of the second law of thermodynamics. Yeah,
Yeah.
it's nothing. Literally nothing is 100% efficient.
Yeah.
Cells. Next, Limosa laponica can fly 9 days straight with no stops to rest or sleep. 11,680 kilometers, weighs 10 ounces. We can't make anything today so energy efficient. Next, the human brain operates at around 25 watts. A comparable silicon-based machine would require 10 million watts. The brain operates at the...
Is this a little meme down here?
I just want to point out the meme in the bottom,
Yeah.
just the meme cameo. You can't see the whole meme, but I just want to point out the meme cameo. Again, folks, this is theoretically a scientific talk. I don't know what we're doing. I don't know what we're doing here.
Yeah, I'm not anti putting a fun little meme in your talk to just sort of lighten the mood, but generally, it needs to have a purpose. It needs to be like relevant to what you're talking. And usually people put it in at the very end on like their acknowledgement slide in like a silly little way.
Right.
But yeah,
it's either gonna be at the beginning or the end. It's not gonna be while you're making your argument.
Yeah,
and you're not gonna cut off the punchline because I don't know what this meme is showing.
Yeah, that's true. He's not even showing this meme. He's like screenshotting a web page that happens to have a meme on it.
Crop your pictures. Just crop them. You don't have to.
Extreme limit of thermodynamic efficiency defined by the Landauer principle. Therefore, human designs will never do better. Next, enzymes are biocatalysts that operate with catalytic efficiency better than anything we can build from scratch, especially enzymes like topoisomerase, which I happen to have published on. Sharks can sense electric fields generated in the muscles of its prey at the limit of what is physically possible.
Can we just go back to that shark thing for just a second? Because this is another one that is, when I saw this, because he's blown by these real quick, right?
We are dish galloping. Yeah.
And I just want to, just for everyone else, kind of another kind of rule, guideline, best practices for doing a talk like this. If you show something on screen, you need to explain it, right? If you show a graph, the first thing you do is say, this is a figure that shows the relationship between what's on the y-axis and what's on the x-axis, and you define what those variables are, right? And thank you, Darren, exactly. If you show a picture that has words on it, you need to either those words better be plain, whatever language you're presenting in, so everyone understands it, or you need to, if it's a technical term, if it's a variable, if it's an equation, you need to define all the terms, right? Or if it's in a different language, you either need to remove the words or define the words, right? I have no idea what the purpose of this is other than complex, therefore something, right?
Yeah.
Because I don't know what this is showing, because it was neither explained nor the French. I believe that's French. I don't speak French. I believe that's French.
Looks French to me.
It wasn't translated. So I don't know what this is actually showing for us, right? So just best practices when you are doing, again, a really tight, concise conference talk, any data you show, you got to clearly explain. Any figures you show, the purpose has to be crystal clear. And part of the purpose being crystal clear is don't show words in another language that you don't translate and explain.
Yeah. And I just want to I am so lost. Like in my notes, I wrote like a 6-minute nothing burger in the middle of this, because it's just I don't know how any of this is connected to fitness. Right? Like he's just going through complex systems and saying, here's a bunch of complex stuff that I guess is arguing is that I guess we're eventually going to get to, they're at their biophysical limit or something.
So you can't increase fitness. Yeah, right. That's the idea. And what I wrote here for this, there's a big chunk in my notes where it just jumps 2 minutes. And the note I have for that approximately 2-minute interval is all the examples of natural engineering. What's the point? There's no therefore to any of this.
Yeah. It's just very unclear what he wants us to take away from this. Yeah.
Thank you,
limit of
chat,
what is
for helping us
physically
translate the French.
Oh yeah, we're getting through here.
possible. The eye can detect a single photon, the nose single molecules. Therefore, human designs cannot do better as a matter of principle. Natalie Angier, after imbuing
This is kind of like yeah. So him going through this whole argument about how there's all this perfectly designed things. I'm just immediately what comes to my mind is all of those gazillion like experimental evolution studies where they basically take a naturally occurring protein and then they iterate through all the possible amino acids that you could replace with, and then they actually generate like Andreas Wagner has done some really cool work on this where they can generate in effect an empirical fitness landscape and then see that actually most existing like enzymes in nature could be improved. Like if they could just get through the mutational pathway to get to a different peak, they could be better than what nature has designed. So like it's very hard for me to think that any of this stuff is like perfect given what we know from like molecular biology.
And to give people like a concrete example of that, Taq polymerase that we use for PCR, right? This is TAC, stands for Thermus aquaticus. It's abbreviation for the organism that we isolated this high thermostability DNA polymerase from, this enzyme, and we use it to amplify DNA in the lab. Now, everybody that does PCR describes the enzyme that we just call it TAC or Taq polymerase, but nobody uses the actual like natural form of Taq polymerase. We've improved upon that natural enzyme to make it work with higher fidelity, to make it work with, to make it more processive, to make it have longer runs before it releases the DNA. And I'll give you a concrete example of why this matters. When I was in grad school, I was doing a lot of this on circular viral genomes that were approximately 5.5 kb. So they were 5,500 approximately single-stranded DNA nucleotides. And I would have to amplify these genomes. And with an improved Taq polymerase, I could do it in 3 overlapping chunks because each enzyme would cover approximately 2,000 bases as it went. Now, the natural Taq polymerase will not run for 2,000 bases straight. It'll run for, I don't know, high hundreds to low 1,000s, right? So I would have to do 6 PCR reactions or 7 to get to fully amplify that genome. But with the improved enzyme, I was able to do it in 3 reactions, which let me tell you how much work that saved me in grad school, right? That cut my PCR time in half. And I know Zach will appreciate how much effort that saved me. And my point here is this: there is a lot of room for improvement in that enzyme in nature to make it work better across a bunch of different metrics than it does. But those mutations that would allow it to do that don't actually help the organism. So the fitness in nature of using the improved version versus the natural version, that difference is not so great that you're going to see a directional shift towards the improved version, right? So it shows that we talk about like the limits of like natural selection and fitness. It illustrates this very important aspect of natural selection, where once you're good enough, you're good enough, and there's a limit beyond which the difficulty of getting the biophysical improvements, the payoff in terms of the representation of your alleles in the next generation, that math doesn't check out. So there's actually no selection for those better variants. But a lot of the stuff in nature is like that, where it works as well as we need it to. In theory, your spherical chicken in a vacuum context, you can make it work better, but in nature, those improvements don't matter. That's the case for a lot of biology.
Yeah, there's also this like kind of issue in that you often arrive at whatever the nearest fitness peak is on the adaptive landscape, and that's not necessarily the highest peak on the landscape. And actually, mutations are going to walk you to whichever is the most mutationally available is the kind of way to think about it, because the way genomes are structured, right, they're physical things, and so certain parts of it are just more mutable than others, just based on like biochemistry. And if those mutations get you to a near but smaller fitness peak faster then like, harder to get mutations could get you to the bigger one. Your selection is going to take you to the first peak, right? And so
go ahead.
So yeah, so just the combination of like the mutation bias, the fact that mutations are biased in the way they occur across the genome, and that selection is very short-sighted, right? Selection is just like what's good now, and this is the best I can get to right now, and then you get on this little local optima, and then you just can't make the leap to get to the
folks, you can visualize it this way. If you have a steep slope, right? So very few mutations, a steep slope represents few mutations. If you have a steep slope to a high but not optimal peak, you're going to get stuck there, as opposed to a shallow slope to a higher peak, right? Shallow slope means it takes more mutations to get there. Even if the peak, the fitness, is ultimately higher, if it takes more steps to get there, you're more likely to achieve the steeper slope, lower peak first, which is still going to be high fitness, but not optimal. And you can, again, there's lots of math and modeling you could do to show this graphically, but that conceptually, that's what we're talking about here.
Yeah.
Interviewing several biophysicists said that biological systems couldn't get faster, more sensitive, or more efficient without first relocating to an alternate universe with alternate physical constants.
We love to hear what physicists have to say. They're really cute. I like physicists. They seem like nice people. They're really cute. I don't care what they have to say about biology. I'm sorry. I really don't care what a physicist thinks about biology.
Emmanuel Todorov at University of Washington mused, "You might say, well, the human body is sloppy, but no, we're better designed than any robot."
Can we just Zach, do you think we could set up a thing for Evolution 2026 where everyone makes when you register and there's a fee to register and attend the conference in person? Can we just be optional donation to anytime a physicist wants to opine about evolution, we just give them $5 instead and they keep their mouth shut? Can we have that? Because I'm with you. I don't care what physicists think of my field. And honestly, I would ask that they maybe, learn a little bit about it before spouting off.
Yeah, there's this great paper that Michael Lynch just put out. I'm forgetting the name of it now. It's like the complexification and the false utilization of evolution. Hold on, I've got to pull it up. It's such a good paper.
It's so good.
It's so good.
The abstract is just...
complexity myths and the misappropriation of evolutionary theory. And it's basically just a broadside against physicists and chemists and just everybody that has been just spouting off about evolution for decades. And Michael Lynch is guys, get out of our field. It's
Stop.
if you want to, and you can contribute to evolution, but boy, you've got to know what we're doing. You've got to understand the principles of our field before you're going to come in and just change it.
It's the XKCD with a race car in a train thing with experts in a different field coming into one field and thinking they've overturned that field. It's exactly that XKCD comic.
Yeah.
This also agrees with the work of Stuart Burgess, professor of biomechanics and robotics for 40 years, who incorporated award-winning biologically inspired designs into robots and spacecrafts. And the final frontier of excellence in biological systems is in the area of self-assembly, self-healing, and von Neumann self-replicating automata, areas in which human engineers cannot even begin to compete. I worked in biomolecular systems, and self-healing is just a nasty problem we haven't solved, but it's solved in biology. Nobel Prize winner Sidney Brenner said von Neumann's automata should be central to biological theory. The best example of Turing and von Neumann's machines are to be found in biology. Nowhere else are there such complicated systems. Code script must form the kernel of biological theory.
Hey, Zach, can we just pause right here? And let me just ask you, how many minutes into the 13 minutes are we on this talk?
We are now almost 8 minutes.
And we have seen zero data about anything.
Yes, we have seen no data. We have not really even seen any graphs of data or any plots of data, right?
It's just a lot of Wikipedia headers and abstracts being quoted.
Yeah, it's pretty strange. And a couple of pictures from other papers, a New York Times article, right? It's
there was a random WordPress blog in there.
Yeah, it's been a wild ride. just on the very last thing, he was talking about Sidney Brenner and Life's Code. Sidney Brenner is like a super important molecular biologist, right? Like he is one of the co-founders of the genetic code. And so I just wanted to like put the like what he's talking about here is that he actually discovered like how proteins are coded and then like deciphered it as if it was a code, right? And there was this whole push in the 60s and 70s to start thinking about like codes in biological systems, and particularly in the genome. But Sidney Brenner was also a very, very large proponent of junk DNA, and was very aware that genomes are parasitized by selfish elements. So he would have never argued that the genome in its entirety is literally a code, but rather that certain portions of it can be analogized and act like a code. And it's a pretty powerful analogy, especially to sort of get the idea across. But yeah, not that he thought that life was literally a code. Okay, so yeah, we are 8 minutes into Sal's 13-minute talk, and we are now returning to the question and answer that he is going to provide.
Could have been like the second slide after the title and name. This could have been the starting point right here.
Yeah.
So, pause. How do biophysical benchmarks, as just illustrated, relate to the notions of fitness and adaptation? This is really the central question of my presentation today. These physical benchmarks define the maximum possible level of fitness and adaptation in the engineering sense. The conflict between the prevailing definition of fitness in population genetics versus the engineering definition of fitness from physics came to a head when I worked on the problem of fitness maximization, especially while studying Grodwall's extraordinarily compelling paper, "The Theory Was Beautiful Indeed: Rise, Fall, and Circulation of Maximizing Methods in Population Genetics." Nowhere in my reading of this historical magnum opus was there any mention of biophysical benchmarks used in determining maximum possible fitness. Yet obviously the Bird quantum magnetic compass has maximized its performance, dare I say fitness, in magnetic sensing. Same for excitons and photosynthesis, energy efficiency in the human brain, etc.
Okay, so
yeah, go.
Yeah, just to give like a little context about this paper, which I think is a really good paper, and people should read it because it's a fun read. It's published in the Journal of the History of Biology. It's a history paper, and it's actually really fun. And I actually read it after Sal presented this, and was glad I did. I really enjoyed it. But what it's about specifically is about the idea that mean population fitness can be maximized in a population. I was gonna like do the math on this, but I will hold back on I will restrain myself in the interest of time.
We already met our whiteboard quota for the video.
We met our whiteboard quota, so I'm gonna hold back and not do the fitness maximization math, but I'm just gonna talk through it really quickly. The idea in the 1920s and 30s was basically it sort of started with Sewell Wright, but R.A. Fisher also had this kind of idea that you could effectively maximize mean population fitness, and that the idea of adaptation was in effect a maximizing of mean population fitness. And their idea of mean population fitness was intricately linked to population growth rates. So the idea is that if the population is growing in size, that's a sign of adaptation. If the population is expanding and growing, you're increasing the mean fitness in the population. And this worked really well in single locus models. So if you're just looking at one gene and you have frequency independent selection, that is where the fitness is independent of the frequency of the allele that's being selected, then you can maximize fitness, and it works super, super well. The problem emerges when you have frequency dependent selection. So like when fitness becomes a function of the allele frequency itself, you actually end up in these sort of troughs where you can't quite actually maximize fitness. And this problem becomes even more pernicious when you start adding in multiple loci that then interact with one another epistatically. And so all of this sort of came to a head in the 1950s and 60s with the work of some mathematicians like Pat Moran and various others. And they effectively showed that you cannot, using like single locus models, maximize fitness. So that was sort of the fall of the maximizing fitness ideas. However, there's a really fun twist that I wish Sal had talked about that this paper actually talks about. And that is that in quantitative genetics, which has long dealt with multiple loci, whereas, like population genetics often is like 1 or 2 loci, in quantitative genetics, the maximizing principles actually still work really well because the theory has already integrated things like epistasis and dominance and multiple loci. And because of that, they actually do have really robust maximizing principles. And so to be clear, all of this is to say that actually the maximization ideas in evolution are still quite robust, and they're used in quantitative genetics, even if they're not used in the way that like Sol Wright and R.A. Fisher had used them in the 20s and 30s. So there's the context and the history in a really brief nutshell without my whiteboard math of this particular paper.
I'll just add to that, the quantitative genetics that you could do on a whiteboard for this is super cool, and I love doing it in class. It's so much fun when you get through it all and get to the end and it all clicks into place. It's like getting like 4 rows at once in Tetris. It's really beautiful.
It is. Quantitative genetics is really cool. It's kind of an unfortunate thing in that there was this kind of schism during the modern synthesis where everybody was focusing on like 1 and 2 locus models, and that was the big focus. And then over here, QuantGen sort of like went in its own way, and like animal breeders and farmers and stuff were using it. But like
That's
evolutionary biologists,
I love it.
The like evolutionary biologists just like forgot about it until like the 70s. And when like Russell Landy and Steve Arnold came back around, and they were like, guys, we need to be reading these like animal breeder textbooks. Like these guys have got it figured out. And then they brought it all back together, and what was, you could almost call like a second synthesis in like the 70s.
Yeah, it's like people breeding cows, figuring out how you can't just look at one loci for whatever trait it is you want to select for in your dairy cows. It was
really incredible. Like Alan Robertson in 1966 discovered that you can treat selection as a covariance. Like selection itself is a covariance. Like that is such a wild thing. I'm going to do a whole video. You'll see.
It wraps your brain into pretzels and then untangles it in just a beautiful way. It's so, QuantGen is so cool.
It is really, really cool. I should do more QuantGen videos. So I want to thanks for the super chat from My Unfinished Journey. 666. That's a great number. Hail Nero, hail Dr. Zach and Dan, hell holding creationists accountable. Indeed. All right, let's Lush's 1937 Breeders' Equation. This is from his I think animal breeding program. Correct me if that's not I believe that's the name of the book. But Jay Lush is like the QuantGen guy, right? That was like studying studied Sol Wright and like brought a lot of this to animal breeding. All right, here we go.
Same for excitons and photosynthesis, energy efficiency in the human brain, etc. So how do engineers determine if something is adapted to fly? If one asked an engineer how a bird or butterfly is adapted to fly, he would say it is adapted to fly because its parts and organizations are consistent with engineering principles of flight. Basically, for something to be adapted to fly, its wings must be able to generate enough lift to overcome the force of gravity. Its engines must create enough power to enable the airplane to overcome the drag caused by the air, so its wings can generate enough lift, etc. That illustrates the engineering view of the meaning of adaptation, and unfortunately it doesn't at all involve adaptation by appealing to reproductive efficiency.
So this is very silly, and this just like completely like evacuates any meaning of the word adaptation.
If you asked an engineer what adaptation means, it's like, why don't you just pick some random word from physics that I don't know and ask me what it means?
Yeah.
I'll give you an answer based on, like, if you take a random word in like physics or chemistry or I don't care, sociology, psychology, linguistics, like pick a field that I am not an expert in, right? Take a word from that field that also has a meaning in everyday English and ask me what that word means, and I will give you a wrong answer. That's easy. Like, okay, I don't know what that demonstrates about the field that you asked me about.
Yeah, it's also just like completely useless, right? Like if the definition of being adapted to flight is the capability of like conforming to the engineering principles of flight, then planes are adapted to flight, right? Like that's like helicopters are adapted to flight. Like that's dumb. Like that's not what we mean when we talk about adaptation, and it just completely voids the meaning of any of these words. So there's a reason that we don't use that kind of definition, is it doesn't actually allow us to predict anything, and it's not explanatory of anything.
From numerous experiments, it now appears the engineering viewpoint of fitness is anti-correlated with the population genetic viewpoint of fitness. These experiments serve as counterexamples to the ultra-Darwinian viewpoint as expressed in popular books, such as The Blind Watchmaker.
So do you see what he did there? He said Darwinian at first and then like corrected it to ultra-Darwinian. And I wonder if that was him like trying to like not sound like he was critiquing evolution necessarily itself. And so he just like plugged in the ultra in front of it. Yeah.
And Darwin's dangerous idea. Ironically, Darwin's dangerous idea advocated the view that biology is engineering. And if we want to get more engineers involved, this is the way it's going to have to go, but they have lots of money. If I may point out an important aside, Wolf and Koonin wrote, quantitatively, the evolution of genomes appears to be dominated by reduction and simplification, punctuated by episodes of complexification. Punctuated complexification. I like that idea, but unfortunately, it may not be subject to repeatable experiments.
Okay, this is another paper that he does all the freaking time, and it drives me absolutely crazy. I'm just gonna I'll just share my screen really quickly to show this paper, because I think it will be useful to show just one of their figures in this paper. So this is actually a kind of a think piece paper. It's a sort of it's not philosophy, but it is more of like a big ideas paper in biology. It's titled "Genome Reduction as the Dominant Mode of Evolution." But
yeah. And Zach, if you go back up to that heading real fast, just go all the way to the top, and everyone can see prospects and overviews, and then on the side, problems and paradigms, right? Yeah. So this is not like a primary research article. This is a big kind of synthesis type piece.
Yeah, yeah. They're trying to put forward some big ideas, and I want to show you this figure because I think this is a really helpful way of viewing what they're trying to argue. Now, imagine, for example, the complexification, and they're specifically modeling genome complexification here. That's really important to note. So the y-axis is complexity, and then the x-axis here is time. And what they're basically arguing in this paper is that a lot of like the big complexification in evolution happens by like whole genome duplications, right? So you get in one event, you get a whole genome duplication that now suddenly you've got a lot more stuff to work with. You get a lot more genes that you can now tinker with and play with and like neo-functionalize, etc. But then you also have a lot of junk that has to get purged, right? A lot of that stuff is just going to become pseudogenes and is going to become nonsense. But it takes a long time to gradually delete that stuff.
Because deletions are not deterministic or purposeful. They're probabilistic processes. And the whole thing is if you delete a region that has been duplicated and one of the two copies still exists doing its original job, then there's no fitness cost to the deletion. Whereas there is a small, but in certain contexts, detectable fitness cost just having that extra crap in your genome. So on net, you're going to see over time both neutral and selective processes to reduce the size of the genome by getting rid of all this duplicative or non-functional stuff that's in there.
Yeah. And so... Yeah, but the thing is, right, is that the duplication, the complexification happens really quickly. It's one mutation, it's one duplication. So you get this rapid increase in complexification and then this long time of like sort of like getting rid of the junk that didn't get neo-functionalized. And then you get another duplication of really rapid change, long. And so the dominant mode, as you can see here, is reduction, right? The dominant mode over time is trying to get rid of stuff. And then you get this quick burst of complexification during duplication events. That's what they're arguing. And so when they're saying, like, when they have this title, you know, like genome reduction as the dominant mode, that's what they mean.
Yeah.
Is that duplications happen really fast, and then it takes a long time to delete the nonsense.
And you would know that if you read this paper, and I'm convinced not one single creationist who has ever cited this paper has read past the abstract.
Yeah, no, definitely not. Yeah, so we've got another super chat. Strawberry, 5th super chat. Are you the 5th super chat? Is that true? How do you know that? How can you
I think that's I think it's just like some kind of inside joke in his name.
Okay.
But now it has changed.
Yeah, Charles, quote unquote, Ultra Darwin. Yes, that is indeed Charles's middle name. It's very funny. Okay, so yeah, so I just and I do this again because Sal shows this paper all the freaking time. It drives me crazy. It has nothing to do with what he says it has, and it's super easy to see that like that's not what they're talking about.
Can we just, before we move on, and I know we're almost done with this talk, we're getting there, but I also just want to point out that what goes on here is related to another thing that creationists do, which is when they take the one line from the abstract of the paper, there's always that one line that is like, hi, everybody, this is an unsolved problem. Hi, everybody, so far we haven't addressed X. Hi, everybody, nobody knows how Y happened. And that's like within the first couple sentences of the abstract. And then if you read the rest of the paragraph, it says, we designed a novel approach to test blah, 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 blah. We determined that blah, 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 finally addressing blah, 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 unsolved problem, right? And then creationists cite that first part where the authors are saying the problem as like, look, Eugene Koonin says this is a problem in evolutionary biology, ignoring the fact that at the end of the paragraph, they say how they solve the problem, right? Yeah. This is kind of a related thing going on with this paper where they literally, the most cited part of this paper among creationists is just the title.
Yeah.
And they don't even, there's nothing about the rest of it. And they just think like we're good, but they don't actually see what the paper says about why we see the trends we do in genome evolution.
Yeah, and again, I think it's a great paper. I think it's a paper you should read. I think it's readable. It's like I like big question papers. I think they're really fun. So yeah, it's a great paper. It's unfortunate that it's been just like completely evacuated of meaning by creationists.
That said, the idea of rapid punctuated complexification may be supported by the idea that proteins are instantiations of Platonic forms. Thus, there may.
Oh my gosh, the Platonic.
This part killed me. This part killed me.
The Platonic forms of proteins.
Like, there's like however long this part goes on, the Platonic ideal of blah, 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 whatever. Yeah. This part killed me.
Yeah.
There may be no gradualistic Darwinian route to evolving some of these proteins. When I look at amino acid residues of collagen on the left and human zinc finger 136 on the right, and colorize the residues, it suggests to me there is no common ancestry from a single ancestral gene locus for all proteins, nor gradual stepwise route for their emergence. This problem would seem even more acute for proteins whose function is critically dependent on multimeric quaternary structure, like topoisomerase, thus reinforcing the notion that proteins instantiate Platonic forms.
Yeah, this is real fun, right? This is really fun. I love that he managed to get his like protein orchard thing into the talk, right? Like it was a nice little Easter egg for those of us that have been like listening to Sal for a while. So that's fun. And he even stepped it up, right? He not only got it in, it's now a Platonic form, which is just wild to me.
Right. We're not even talking about fitness maximalization or whatever anymore. It is a Platonic ideal.
It is a Platonic. Like, and when he said it, I literally had to be like, am I confused on what Platonic forms are? And I literally like went back to the Wikipedia page to like brush up to make sure I wasn't crazy. And yes, indeed, it's just like this ideal thing in which all of reality is merely a mimic of the ideal, perfect, abstract entity. And this is just like really bizarre to me to think about a protein as being a Platonic form, especially given that proteins have like vast families, and like most of them very clearly, like each individual like protein family like has ancestry within itself, and we see proteins emerge all the time, right? Like we have so many studies of like de novo protein emergence.
Yeah.
So unless Platonic forms can just poof into existence, like that just doesn't make any sense to me. It's just absolutely wild.
Additionally, consider this experiment by von Hofwegen et al., which showed that the DCSU protein in long-term evolution experiment never recovered function over 80,000 generations following a mere 5 nucleotide deletion event. This experiment supports the anti-correlation hypothesis, but I would hope for hundreds more of such experiments. In some cases of reductive evolution, it may be powered by natural selection, as in this study. Again, reinforcing the anti-correlation hypothesis.
Zach, can you just go back to that for a second? Can we just appreciate this? Can we just appreciate this for a second? More intelligent men and women. This is like some Evo psych nonsense going on right here.
This
and
is. Yes.
I just like every paper that you choose to cite to support a point is a choice. And the fact that Sal is using this is clearly some EvoPsych trash right here. And the fact that he picked this one from Social Science Research, that's the journal, to support his point here is, man, that is a choice.
It
right?
is. It
Like,
is.
just everyone read those, like the highlights. Read the highlights. And I want to be clear about this. These are the highlights that the author said, not somebody making fun of this paper said.
Yeah. Yeah, this is the kind of like absolute nonsense that is embarrassing that gets published at all. And if you go in and you look at like the places that have cited this paper, they're in journals like like Intelligence, and like Mankind Quarterly, and like these like very obviously like racist pseudoscience, hereditarian nonsense journals. Like, yeah, I mean, this
It's bad.
I it didn't have much on this Wikipedia page, so I didn't have time to do like a lot of digging on this particular journal. It's got a pretty low impact factor, so I don't know how often like
I don't look. I'm not judging the journal. I'm judging the paper.
Sure.
And this is EvoPsych dumpster fire. Right
It is.
here. Like straight up.
Yeah. We do have another super chat here from my unfinished sock for $2. Don't miss the point of evolution. Like and share. Thank you so much, my unfinished sock.
This study, again, reinforcing the anti-correlation hypothesis. Even more pointedly, the famous long-term evolution experiment shows anti-correlation between engineering fitness and population genetic fitness.
Yeah.
God damn it, Sal. Like, there's basic population genetics to explain this. When you have more mutations, you sample more sequence space, which means you're more likely to find the optimal genotypes. You're also more likely to mutate away from those genotypes. So what you get, and this has been documented in bacteria and in viruses, is that in a high mutation rate environment, you get a higher fitness maximum, but you get a lower fitness average. And my friend, there is very simple, and it's math that's easy enough that I can follow it, math that explains this dynamic. You see this in the Lenski mutator line. You see this in the Springman 2010 paper in bacteriophages. It's the same dynamic where if you sample more of sequence space via an elevated mutation rate, you both find higher peaks, but you mutate away from those peaks faster, and the average fitness goes down, even though the maximum fitness goes up. This is not hard to explain at all, but this, talk about dog whistles, this is Sal doing a genetic entropy dog whistle is what's going on here. And he's not saying those words, but that's the argument he's making.
Yeah. Yeah, he's definitely making this argument, and this is actually such a cool paper. They found something
awesome.
that's like really, really incredible. And to just like, and again, this is one that he shows all the freaking time. And so I just want to like explain really quickly like what they did or what they were trying to test. They basically were comparing, and I'll just go ahead and I'll pull it up. They were basically comparing. They were comparing a couple of different strains. And you basically have these mutator genomes. And in case you're unaware, mutator genomes are those in which the enzymes for repairing mutations are basically broken. So you're just getting a lot more mutations in them on average than you normally would. And what they were interested in is comparing how natural selection is going to operate in those genomes versus how they operate in genomes in nature.
So beautiful.
And how they operate in genomes in mutation accumulation experiments. So these are the three different plots. We're only really concerned with this very top line here. I know it's maybe a little hard to see. Let me scroll in just a little bit. Okay, this very top line. What this top bar represents is the ratio of synonymous to non-synonymous mutations, okay? When you have more synonymous mutations than you do non-synonymous, that is an indication of purifying selection. So basically, natural selection is purging all the non-synonymous mutations that would otherwise be changing the protein. And so whenever you see this like green over here to the right, that's showing that the ratio is much higher of synonymous than non-synonymous. So in nature, again, they had this natural test. In nature, they saw way more synonymous than non-synonymous, indicating that natural populations of bacteria are undergoing strong purifying selection. But then look at what happens in this one. This is the mutation accumulation experiments. In those, what you're trying to do is remove the action of natural selection. You want the mutations to be able to accumulate so you can see what they do, right? So what you do is you isolate like a single bacterial cell, and then you allow it to colonize the dish, and then you can just calculate all the different mutations that occur without the inhibition of selection, right? And so you can see the line is almost invisible here. It's like right near zero. So you're getting the same amount of synonymous as non-synonymous mutations. That's what you would expect if selection is inefficient at getting rid of those deleterious non-synonymous mutations. And that's what you expect if like genetic drift is driving the dynamics. Right?
And the reason, and Zach, just to go one step further for everybody, the reason for this is that the way you design these experiments is you put the population through a bottleneck. So every iteration, you start with a very small population, which means very low competition, so that there's so many resources available relative to the demand from the population that exists. There is lots of room for suboptimal genotypes to still persist in that population. That's, you design it that way.
You want the mutations to accumulate so that you can see them, you can measure their effects, etc. Then you have this other one. This is the long-term evolution mutator strain. Now, what's really cool about this is they didn't go through bottlenecks, right? There's no bottlenecking here. And yet, look, it's actually almost indistinguishable from the mutation accumulation. Why is that? So what they're testing here is what's known as genetic draft. And this is an analog to genetic drift, but it's driven by selection. And this is a really, really cool idea. So in bacteria don't undergo recombination. That's not strictly speaking true in nature.
It's more true than, whatever.
In the lab setting, bacteria typically don't undergo recombination. And because of that, if you have a genome that is now getting a crap ton of mutations all across it, right? It's getting so many more mutations that now it's getting a bunch of beneficial and a lot of deleterious, right? So you're getting a whole bunch of mutations. But so long as the net effect of the beneficial is greater than zero, so long as it's just above the deleterious effect, selection can grab it and it will sweep all the mutations to fixation. So all the good, all the bad gets swept to fixation all at once. And so what this looks like is it just looks like genetic drift is dominating it. But the problem is since there's no recombination, those deleterious mutations can't get off the good background. And so you're always sweeping every, bad mutation along with every single good one. And and they call that genetic draft because in effect what's happening is selection itself is interfering with its own ability to purge deleterious mutations because it's favoring the good ones. So it's like a reduction in the efficiency of selection, like what drift is, but since it's selection itself, they call it draft. So to me, this was like such a cool paper that struck at like the heart of a really, really neat population genetic idea. And for Sal to like be misrepresenting it like so badly, it just drives me crazy. So yeah, the reason that these two look so similar is because of linkage. It's because of selection and linkage. Now, it is important to note that in nature, bacteria do undergo recombination. And in fact, they undergo just as much recombination as we do. They just do it differently, right? They do it through horizontal transfer. And this is why you can still get really strong purifying selection in nature, and why you can still get lots of adaptation in nature without having to worry about linkage in the same way that you normally would, because you do still get a lot of recombination. So that's just a rundown, because again, this is a cool paper. This is a really cool paper that people should read. They did something really, really neat. This is not in any way a genetic entropy story at all. This is like one of the first, as far as I know, one of the first like big demonstrations of like genetic draft, right? So I think this is super
really cool.
Yeah.
I don't have anything to add. That was a fantastic explanation, and this paper is super cool. I love this stuff. I love it. I get to teach it a couple times a year, and I just love it so much.
Yeah, super cool.
I start drawing graphs and like intersecting curves on the board, and my students maybe not as much as I do, but I really love it.
Yeah.
Like, look, see where the lines cross? That's where,
Yeah.
And population genetic fitness. Cooper and Lenski showed fitness increased in one environment and declined in multiple and other environments that tested catabolic.
Just antagonistic pleiotropy. Stop the presses. like as though nobody knows what this is.
Yeah.
Everybody at the evolution conference knows exactly what phenomenon explains this.
Yeah. Yeah. And this is also like in many ways kind of a basic ecological principle of trade-offs, right? Like this is the thing I would expect people to learn from the very beginning, is that nothing is perfect in every environment. Everything exists as a trade-off. You gain one thing, you've lost something too. You've lost something in a different environment. That's just what trade-offs are.
Look, everybody watching this is, however you classify yourself, a nerd or geek to one degree or another. So many of you will understand the analogy that I like to use in class, which is like if you're doing character creation in an RPG, you can't maximize all your scores, right? If you're going to have 20 for intelligence, then, your strength or constitution is going to take a hit, right? You cannot be good at everything. That's just how it is. And in this experiment, what we're not getting into here is the design of this experiment. The bacteria are specifically given a finite amount of glucose every day, and it runs out every afternoon. So they're going to get really good at absorbing and metabolizing glucose. And if that comes at the cost of becoming less efficient at using some other carbohydrate source of energy that they're never exposed to, then the cost to that trade-off in the experimental environment is zero, because they're never exposed to whatever other sources. They don't get fructose. So if you have enzymes that break down fructose, who cares if if those enzymes mutate in such a way that somehow improves your ability to metabolize glucose, right? That's just how evolutionary trade-offs work. And we call that antagonistic pleiotropy, where there's more than one effect for a particular mutation that occurs. And one or more of those effects are beneficial, and one or more of those effects are harmful to your ability to succeed in your environment. And if the harmful effects never manifest because that's not your environment, doesn't matter.
Yeah. I want to point out, Darren Simmons noticed this, that I didn't even see this when I watched it, but it says here, adapted by Chris Roop. So I didn't know that Sal was here citing Roop. And then look at the way he spelled E. coli. Escherichia coli instead of Escherichia.
And they got the capitalization backwards. That just hurts me.
Yeah, I didn't even notice this before. So that's that's a wild thing.
Efficiency, and this is strong evidence in favor of the anti-correlation hypothesis. And beyond the fact we are now in the 6th great extinction, the biosphere may be in an era of reduction and simplification, to use Wolf and Kuhnen's words. And declining genetic health over time is a testable prediction, at least in principle, that could lend support to the anti-correlation.
Yeah, he had to throw this in.
Got to throw it in there. Michael Lynch, genetic load paper, drink/bingo, whichever game you're playing. There you go.
Yeah. He had to throw this one in. In case you're interested, I have a whole video on this paper. I forget the name of it now. Maybe it's just called the Human Genetic Load or something like that. But basically, that just reviews this paper. Michael Lynch normally hits them out of the park. I'm almost always on Michael Lynch's part, or on Michael Lynch's side with biology. This paper, trash. This paper was trash. And I will say that this paper has been responded to by John Wakely, who is one of the biggest theorists in the field of coalescent theory. So a really, really well-respected population geneticist. So I highly recommend you check out his response to this Lynch paper.
Hypothesis between the engineering perspective of fitness versus the population genetic definition of fitness. But in the meantime, let us all work together to preserve and study the ingenious designs in nature composed of organs of extreme perfection and complications that constitute endless forms.
Just want to say I'm very jealous that you met John. I saw his talk at the last Evolution in Montreal, and it was really cool. I barely could follow it. I have his book on my shelf, and I've read it a few times. It's one of those that you got to read it 100 times to be able to get all the math.
It's beautiful and most wonderful. Thank you. And at the worst possible time.
Yeah. Okay. So he did get a question there at the end. I mean, it was just what are your sources or something that I didn't bother keeping in. But there's the creationist that gave a talk at Evolution. It's a pretty wild thing. I think it's, one, hats off to Sal. Hats off to Sal for being willing to do this. For going to an evolution conference, walking into the lion's den, and being willing to actually to present this stuff. I do want to give one farewell to this idea. This is one of my favorite papers. This is by Nancy Chin. She's a population geneticist at the University of Rochester. Just does absolutely wonderful work. This is in a completely pedigreed natural population of the Florida scrub jay. This is a really, really cool system where they basically have just mapped out every single mating, all of their offspring, and then like who all their offspring mated with across like multiple generations. So it's a fully pedigreed population. So this is super, super cool. And what they do here is they can use population genetic models based on population genetic definitions of fitness and do some really incredible things. And I just want to show you this plot. So this plot here, this is the observed allele frequency change in this population across multiple generations. Again, this is a natural population, and this is what they predicted given their mathematical models. And just look, this is the one-to-one fit. Like, this is an incredible ability to make this prediction, right? This is almost entirely falls along the one-to-one. And I challenge any creationist that has like their own definition of fitness, or that thinks that the evolutionary definition of fitness doesn't work, or that there's some missing mechanisms in evolution, and explain to me how it could be that we could get this accurate of a prediction of allele frequency change through time. I'll give you just one additional graph. This is across the years. The blue is the observed, the black is the one in the model, and then the gray bars are the 95% confidence interval. Again, this is a prediction made by a mathematical model of evolutionary fitness without using any engineering principles or anything like that, and we are able to predict how it would change through time. I don't think Sal's definition could even come close to doing anything even remotely like this.
Yeah, I think Guts Gibbon kind of hit the nail on the head here. The reason they want to change the definition is because the normal one works and supports evolution. Yeah. But we're all afraid of math or something. I don't know.
Yeah. Okay. Yeah. So that was Sal's talk at evolution. Do you have any closing thoughts or any last thing you would like to say?
Yeah. So I want to recommend to everybody that you can find this. You just go on YouTube and put in evolution 2025, and you can find these videos on YouTube. And in Sal's own video on this, there was a bit of like a conspiratorial kind of preface to it about the videos not being uploaded or what. They're all there. You can find them on YouTube. They're there. Like, Sal, I don't know where you're looking. They're there. They're on YouTube. That's how I found it. But that aside, I want to encourage everybody to find that and just check out the other videos in the session that are real, like evolution, pop gen kind of data driven things. And you will see very clearly that what Sal is doing is not what everyone else at this conference is doing, right? We just went through that. There was zero primary data. There was none. There was no primary data here. And the other people are saying, hey, here's a question I'm going to answer, and here are the data. Here's how I did it. Here's graphs and data that I, answer this question. The other thing is that this was in the the population genetics theory session. And like, I suppose that's what this falls into, but like, this is not, it's not really contributing to PopGen so much as saying the way we do PopGen is wrong, right? Because if you're going to dispute like as fundamental an idea as fitness, the way we use it in population genetics, you're basically objecting to the whole way population genetics works. And if you're going to do that, you better come with the goods, because like Zach just showed, we can make extraordinarily accurate predictions with our current understanding of population genetics. So if everything is wrong, why is that the case? Why are we able to do that then? But the really big thing. Oh, go ahead. And then
I was just gonna add to that point that like the goal of population genetics is to be able to predict changes through time. And and we do that by having a mathematical understanding of the mechanisms that propel populations to change through time.
Yeah.
If you think you can do better, then give a model that matches the data better than ours do.
Right.
You don't have it. You just don't have it.
But the last thing, and the thing I really want everybody to take away from this, is Sal Cordova, creationist. Like, he's been doing this for decades, folks. Professional creationist. He had right in there, I'm pursuing a PhD at Liberty University. And I guarantee you there are people in the audience that saw that and went, oh no. He was allowed to present. No one's censoring him. No one's stopping him. There were no questions that were belittling or, making fun of or whatever. The only question he got was, hey, can I have a list of your sources? All the papers that you referenced in there, right?
Yeah.
Okay. Every other professional creationist, what's your excuse? Behe, Carter, I don't know, Douglas Axe over at Discovery Institute, right? Jason Lyle, Nathaniel Jameson. What are you and your Harvard PhD doing? Like, come on. If you're so right, get in the ring and make your case. And somebody earlier was like, well, they're afraid people are going to be mean. Have you seen what scientists say to each other?
They are going to be mean. They're mean to eat. Like, we're mean to each other.
Right. Yeah. Merciless to each other. So you know what? If you can't take the heat, get out of the kitchen. But yes, people will challenge you on your ideas. You're not going to be censored. You're going to be held to the same standard of evidence as everybody else at the conference. And if people don't like your ideas, they're going to tell you and they're going to give you a reason why. And they're not going to call you a creationist poopy head. They're going to tell you that you are fundamentally getting basic population genetics equations wrong. Right.
Yeah.
Do it. Everyone else, do it.
Yeah. It drives me crazy to say that they're afraid or the idea that we evolutionists just walk around and pat each other on the backs and oh yeah, good job.
Right. We're all just everyone makes sure that no one realizes we're all lying. Everyone just keep up the facade. No, absolutely.
It's very often you give a talk and you get hard questions at the end, or you give a poster and you're going to have somebody come and ask you hard questions at your poster. Anytime we have undergraduates and stuff that present at conferences, we always are checking on them. You'll be look, and we always tell them be sure you put that you're an undergrad, and that makes people go a little bit softer on you,
a little less. Yeah.
They don't come at you as hard, but every year there are people that get their feelings hurt at these conferences. Okay. So, and it's because people are ruthless. So I don't want to I don't want you to think that we just baby each other or that we just sit around and don't challenge each other. Scientists they like to argue. Heck, that's why we do this, right? We do this because we like to argue. We're just finding more people to argue with.
Right.
And so this is what we do. We like to do it. So if you don't be afraid, come to it. And then if you stand on the strengths of your convictions, and let's see what you got. Okay.
Exactly that.
Yeah. So I think that's we're approaching the 2-hour mark. I think we actually hit the time pretty well here. It only took us 2 hours to review 13 minutes. I think that's pretty good, right?
We had a time constraint and we hit it, and we didn't have to rush through anything. We hit our marks because because Zach, we practiced this very precisely.
Yeah, we did. Okay. So tell the audience before we go what you've got coming up on your channel on your channel next. What can
Thank you. Why do you have to put me on the spot like that, assuming I have things planned? Because I will tell my audience I just finished a 4-week summer course, so I will come up with things for the next few weeks over the next few days to ensure I don't miss anything. I do have a few things. I recently appeared with Rebecca, Bread of Life, on her channel, Examining Origins. We did some very bare bones, genetics, just gaming out common descent versus common design stuff. And that was a really fun conversation. I thought it was fun because it was a really cool illustration of let's walk through this admittedly oversimplistic model of how this stuff shakes out. And just whatever questions your kind of non-specialist creationist is going to come up with as we go, I'm just going to field those questions as we kind of walk through things, and I have answers to them, but there are questions and I'm happy to do it. So I'm going to rebroadcast that at some point. I also, for those of you following the James Tor Rob Stadler saga, So they responded to me and Erica and Professor Dave, and we recently recorded our response. So I'm excited for that. It's and you'll see, but it's notable what they chose to ignore in our response to them and what they focused on. It is what they did not talk about was as notable as what they did talk about. So we'll each be releasing that on our respective channels as soon as we each get around to, editing it and whatnot. And I have some other stuff that's in the works. There's a bunch of videos I've been alluding to for a while that are anywhere from like 10% to 50% done. There's some things that like I just have to sit down and record it. And there are other things that I have an idea and it's basically 2 sentences right now and I have to figure it out. But I do have some fun stuff coming up that I think people will appreciate once I like have time to sit down and actually do it.
Yeah.
So that's what I'm up to. Yeah, there's stuff. It's happening. I hope.
Nice. Yeah, so I've got a really busy summer. I'm actually moving. I'm moving to Georgia to start a new job. I don't guess I've said this on the channel, but I'm going to be an assistant professor at Augusta University in Georgia. So I got a job. I'm not going to be a lowly postdoc anymore. So I'm really excited about that.
Congrats. Congrats. Way. That's awesome.
Thank you. Yeah. Been on the job market for a couple of years. So yeah, it's nice to have like a permanent job, instead of this like contract, a lab tech work kind of stuff. So I'm pretty excited about that. And, but that means that my summer is like pretty busy trying to get everything moved. I do have one big script that I was kind of inspired. I've been on a quant gen kick here lately, if you haven't noticed. And I really want to do like a really big video on what I'm calling sort of the second synthesis. And I mentioned this actually a little bit earlier, this sort of remerging of like quantitative genetics from like the animal breeding world back into evolutionary theory with the help of people like Russell Landy, people like Stephen Arnold, Nick Barton, and a lot of those other people. And I think that it's kind of a forgotten synthesis that I think is one of the most important in like, recent decades. So I want to sort of talk through the history of that and then also give you some of the fundamental derivations, like where do we get, like what is additive genetic variance? What does that mean? Where does it come from? I've been working on walking through the mathematics of Fisher's 1918 paper, which is the first one that melded the biometric school of like the statistics school with Mendelian genetics. Like it was like it's like the paper that founded quantitative genetics. And I'm going to walk through the math for you.
Zach, the people are clamoring for it. You have to give the people what they want.
Yeah.
Will there be a whiteboard?
There will be a whiteboard. In fact, the little bit of whiteboarding that you saw here today was like, I bought all of the things to do this for this video so that I could do this video with the little pin. So yeah, I'm really excited about it. I think it's going to be super fun. It's going to be math heavy, but I'm going to section it up where it's like a little bit of fun history and then some math. So like if you can't handle the math, you can skip to the next fun history and then math. And so it'll be kind of sandwiched in that way. Yeah, so I'm pretty excited. It's taken me a little bit to write because it's going to be a little beefy, but I think it'll be really fun and I think it'll be really informative. And you're going to hear a lot of characters in evolution that are super important that you probably never heard of before. So I think that's going to be that's the next big thing that I've got. Another super chat from Strawberry, ultra Darwinian vein. We love that so much. $2. Congrats on the job. Thank you so much for that. And thank you so much for being here. Yeah, so that's what I've got coming up. Otherwise, the summer might be a little slow because of the moving and stuff. But yeah, well, thank you all so much for being here. Thanks, Dan, for walking through this and giving your feedback. This was a ton of fun.
It was.
Yeah, and I will catch you all next time. Take care.